So guys, PP405 is probably not enough and might fail. And I say the following knowing that
So,
maybe...
did I just come to the conclusion that DHT isn't so much the cause of hair loss as it's the activator of a deeper vulnerability? I guess you can say that. Still, DHT is a significant factor that we can control for, and really, it might be the catalyst that's causing for all of this dysfunction to occur in the first place. But when it comes to this sort of discussion, is it the TRPS1 signaling? What is causing that signaling to become reduced? I just kind of walk away with more questions than I have answers at this point. But one thing matters here, something really important matters here, and that is the occipital donor hair survived in the frontal scalp, just like you would see in cases of androgenetic alopecia hair transplant. The pattern of hair loss and how it occurs in trichorhinophalangeal syndrome is very similar to that of male pattern baldness or androgenetic that being alopecia. The case, they have high DHT signaling to the dermal papilla. That shows us that DHT is causing for these things to shrink, right? Well, at least DHT might have its involvement in those dermal papillas. But it's conceivable, right? We have to think about this because these dermal papillas are smaller in every cycle. So something is happening to it before it becomes a dermal papilla. Something is happening to the mesenchymal stem cells. That's where the focus should be. And we have some very, very groundbreaking literature that we have to string together. Good friend, a good pal of the KWRX Cognita server, and he pointed out some things.
Hello everyone and welcome back to yet another video. Today we're going to be talking about something that I think the hair loss community needs to hear, even if it goes against the current hype. We're going to compare two very different approaches to treating hair loss. PP405 by Pelage Pharmaceuticals, as well as the AMP pipeline products coming from Amplifica, which would include AMP303 being the Osteopontin analog, and AMP601 being the SCUB3 or SCUB3 peptide analog being developed by Amplifica. And I'm going to make the case for why I believe the real path to curing hair loss runs through a tissue layer that most people have never heard of, and this is the mesenchyme. So what is the mesenchyme? Well, your hair follicle is built from two groups of cells working together. The epithelium is the skin side that actually builds the hair fiber. The mesenchyme sits underneath as a layer of supportive, instructive tissue that doesn't make the hair strands directly, but tells the hair building cells what to do, when to start, and how quickly to grow, and also when to stop. The most important mesenchymal structure is a small cluster of specialized cells at the very base of the hair follicle called the dermal papilla, and it functions as the command center for the entire hair growth operation. In androgenetic alopecia, it's this command center and the mesenchymal tissue surrounding it that progressively breaks down. The dermal papilla shrinks and its signals weaken, and then, well, the whole system falls apart cycle after cycle. That is the core of this condition. Not necessarily that the stem cells are sleeping, but they're changing their metabolic activity, and the command center, that is the dermal papilla, well, it's failing. So, with that being said, let's get into it. So guys, PP405 is probably not enough and might fail. And I say the following knowing that maybe I could be wrong. And if I am wrong, that's good because I'll be using PP405 to grow my own hair. However, I think this still needs to be pointed out for the sake of furthering the discussion on hair loss research. And this is that PP405 seems to be largely misaligned in what its goals are ostensibly stated to be. And it might not even provide any significant growth as hair loss treatment press reports will lead you to believe, particularly the claim that it grows hair from slick, bald areas. The foundational rationale for PP405 comes from the observations in normal human and mice hair follicles. During the transition from the telogen to anagen phase, that is the resting to growth phase, there is a spike in lactate production and increased in lactate dehydrogenase activity within the bulge stem cell compartment of the hair follicle. It's been noted that this metabolic shift appears to act as a trigger signal that helps activate hair follicle stem cells and initiate the growth phase. This phenomena is documented in Pelage Pharmaceuticals' foundational research from UCLA, with one paper showing this to be the case in mice hair follicles titled, quote, "Lactate dehydrogenase activity drives hair follicle stem cell activation," unquote. In this paper, we see that lactate dehydrogenase activity is enriched during the telogen to anagen phase transition and shows a signal that increased lactate dehydrogenase activity has occurred in the stem cell bulge area of the hair follicle, and this can be noted by the red staining in the image on the screen. And I'm going to make the case for why I believe the real path to curing hair loss runs through a tissue layer that most people have never heard of, and this is the mesenchyme. So what is the mesenchyme? Well, your hair follicle is built from two groups of cells working together. The epithelium is the skin side that actually builds the hair fiber. The mesenchyme sits underneath as a layer of supportive, instructive tissue that doesn't make the hair strands directly, but tells the hair building cells what to do, when to start, and how quickly to grow, and also when to stop. The most important mesenchymal structure is a small cluster of specialized cells at the very base of the hair follicle called the dermal papilla, and it functions as the command center for the entire hair growth operation. In androgenetic alopecia, it's this command center and the mesenchymal tissue surrounding it that progressively breaks down. The dermal papilla shrinks and its signals weaken, and then, well, the whole system falls apart cycle after cycle. That is the core of this condition. Not necessarily that the stem cells are sleeping, but they're changing their metabolic activity, and the command center, that is the dermal papilla, well, it's failing. So, with that being said, let's get into it. So, PP405 is probably not enough and might fail? And I say the following knowing that maybe I could be wrong, and if I am wrong, that's a good thing because I'll be using PP405 to grow my own hair. However, I think this still needs to be pointed out for the sake of furthering the discussion on hair loss research. And this is that PP405 seems to be largely misaligned in providing any significant growth as a hair loss treatment, as press reports lead you to believe, particularly the claim that it grows hair from slick, bald areas. That's a pretty big claim. When we look at the foundational rationale for PP405, it comes from observations in normal human and mice hair follicles. During the transition from telogen, or the resting phase, to the anagen or growth phase of the hair cycle, there is a spike in lactate production and increased in lactate dehydrogenase activity within the bulge stem cell compartment of the hair follicle. This metabolic shift appears to act as a trigger signal that helps activate hair follicle stem cells and initiate the growth phase. And we can see this phenomena being well documented in Pelage's foundational research, with one paper showing this to be the case in mice hair follicles titled, quote, "Lactate dehydrogenase activity drives hair follicle stem cell activation," unquote. In the paper, we see that lactate dehydrogenase activity is enriched during the telogen to anagen transition phase, and shows a signal that increased lactate dehydrogenase activity has occurred in the stem cell bulge area of the hair follicle, which can be noted by the red stain in the picture. And it's especially interesting because they're doing this with intact skin sections. So they're allowing the hair follicle to sit in its normal structure rather than getting it isolated. And this is important because they have to see all of the intricate structures that might exist between different hair follicles. And like I mentioned previously, they're using this sort of red reaction dye, this colorant agent that reacts with the lactate dehydrogenase activity. And they can essentially, throughout the entire tissue sample, find where lactate dehydrogenase activity is the strongest and where the signal concentrates. So when they isolated the bulge area of the hair follicle where the stem cells are, and also monitored the output of those cells, their metabolites, the researchers noted those hair follicle stem cells in that bulge structure show a glycolysis-heavy signature with higher lactate compared to the rest of the epidermis. And lactate was noted to rise as follicles went from the telogen state into the anagen state. And then that lactate declines once the anagen state is established. So remember, they're going from a resting state to a growth state, and once they're formally in the growth state, the hair follicles decline in lactate activity. Now, increase in lactate means nothing for this particular model if the researchers didn't notice something. So the researchers needed a way to figure out, hey, are the hair follicle stem cells multiplying? And if they are, that could be indicative of some kind of stimulation directly to those hair follicle stem cells. So the researchers decided to track a particular protein called Ki67. Now, Ki67 is known as a proliferation marker. We'll touch on this later in the video, but essentially, whenever a cell multiplies, it releases this particular protein. So now that the researchers have observed that when lactate levels go up in the hair follicle stem cells, the hair follicle stem cells want to multiply. And because of that, the protein Ki67 goes up. So they began using compounds called mitochondrial pyruvate carrier inhibitors. The mitochondrial pyruvate carrier inhibitors essentially block the protein that is responsible for carrying the pyruvate molecule into the mitochondria in order to do ATP synthesis. Now, when you block that particular protein from doing its job, what happens is that there's an excess buildup in pyruvate within the cytoplasm of the cell. Now, remember, mitochondria is known as an organelle, so it's inside of a cell too. And the cytoplasm is the open space of fluid between different organelles within a cell. Kind of like how you as a human being, you have organs within your body, but you also have blood in between those organs, right? And other sort of connective tissue. Anyway, going back to the cellular model, as the pyruvate builds up inside of the cell itself, the cytoplasm area, the cell responds by using backdoor pathways in order to get energy out of that pyruvate that can't be put into the mitochondria. So it increases the enzyme known as lactate dehydrogenase. Lactate dehydrogenase comes in contact with pyruvate and transforms that pyruvate into lactate. Then that lactate is transported into the mitochondria by a transporter protein known as monocarboxylate transporters. So it's clear that the thought process here is that if you can increase that lactate level artificially by using a mitochondrial pyruvate carrier inhibitor, what we call a MPC inhibitor, then you can probably wake up hair follicles that are dormant. And the researchers did a good job because they made this causally related by deleting the genes that make lactate dehydrogenase enzyme, specifically in the bulge stem cells during telogen. And what happened was that most of the hair follicles failed to properly enter into the anagen phase, and then they stay stuck in the dormant telogen phase, creating bald areas on mice. So the Pelage team seems to have made a model that supports the idea that lactate dehydrogenase programming is part of the trigger that kicks off a new hair growth cycle. So that was in mice. What about human beings? Well, in a Dermatology Times interview article published on August 19th, 2024, Pelage's chief medical officer, Dr. Christina Wang, described phase 1 results showing that PP405 increased Ki67 detection in human scalp hair follicle biopsies after 7 days of treatment. Once again, that Ki67 protein is a sign that the cells are actively duplicating, or in other words, proliferating, which seems to be important because, well, that's what PP405 is hinted to be doing. So all in all, PP405 seems to increase hair follicle proliferation activities. Okay, so video's over, right? We should go home, we're done. Well, not necessarily, because although this is meaningful, we need to understand that Ki67 simply increasing doesn't tell you if your hair follicle stem cells are differentiating, or in other words, transforming into the cells that are necessary to grow hair. Again, it doesn't matter if they're multiplying. The crucial step is that these hair follicle stem cells need to differentiate. They need to transform their state, become something new that is important for hair growth. And so far, we don't know if that's the case to any significant extent. So, what are these cells and what do they do? First, we need to be acquainted to the mesenchymal stem cells. When researchers say hair follicles are controlled by the epithelial mesenchymal interactions, they're basically saying that hair growth is a two-team system. One team is in the epithelium, which is the skin and hair fiber side of things. Those are the cells that actually build the hair shaft. And the other team is this thing called the mesenchyme. The mesenchyme is a type of tissue made of supportive instructive cells. It should be noted that they are not directly responsible for making the hair fiber itself. They're more like the planners and coordinators that set up the signaling to eventually direct the hair follicle organ to grow hair fibers. Those hair fibers are governed by another class of cells called keratinocytes. The mesenchyme stem cells sit underneath the skin and they tell the epithelial cells what to do, when to start, when to stop, and how intense the growth should be. In hair follicles, the most important mesenchyme structure is the specialized dermal papilla. The dermal papilla region of the hair follicle is a small cluster of specialized mesenchymal stem cells located at the very base of the hair follicle. It functions as the command center for the hair growth. So, we would call the general tissue structure here the dermal papilla region, but typically people just call it the dermal papilla because it's just easier to say. So really the tissue structure, dermal papilla, is named after the cells that are there and not the other way around. Dermal papilla cells control hair thickness, size, and even color. They help determine how long a hair grows. They also regulate when the hair follicle enters the growth phase or the resting phase, so anagen or telogen. They also help send activation signals to the stem cells in the epithelium as well. And when we say hair follicle stem cells, we need to note that they are not the same as mesenchymal stem cells. Mesenchymal stem cells are from the mesenchyme layer. Hair follicle stem cells are from the epithelial layer. They have different origins. So the area of the hair follicle called the stem cell bulge, well, this is of the mesenchymal lineage. Technically speaking, that means the dermal papilla cells are not of hair follicle stem cell origin. So with that in mind, it's likely that you need to have functioning mesenchymal stem cells that has the ability to transform, also known as differentiate, into dermal papilla cells. And let's remind ourselves again, right, that dermal papilla tissue structure is kind of like a ubiquitous term. You might have different types of dermal papilla that are responsible for different signals, but collectively, they work in tandem to regulate the hair follicle. So the chain of command is actually quite simple. Your dermal papilla cells send out some signals that dictate how a follicle should grow, and it communicates those signals to the epithelial layer of your tissue in the pileosebaceous unit, that is the hair follicle organ. And within that organ area, there is a sub-compartment that's called the stem cell bulge. The stem cell bulge houses these epithelial stem cells that the dermal papilla will be talking to ultimately get them to act different. And they eventually migrate down towards the dermal papilla. They create the various components of the hair follicle, which is the hair shaft, inner and outer root sheaths, just to name a few. So we really need to stress this point across because hair loss is not just about hair follicle stem cells, those epithelial stem cells being active or inactive. It also depends if we can get those mesenchymal stem cells to turn into dermal papilla cells, and if those dermal papilla cells have the capacity to produce instructions to neighboring cells to effectively create a hair follicle. And we'll talk about this later, but maybe there might be something wrong with those mesenchymal stem cells, and therefore something wrong with those dermal papilla cells. My larger point here is that PP405 seems to be focusing on the epithelial side of things, like lactate dehydrogenase related metabolic activity and how it activates hair follicle stem cells, those epithelial stem cells. But really, that's not the same thing as creating or rebuilding the dermal papilla, because, well, they would have mentioned if that was the case. And really, we can see that in how the Pelage team and in their studies, what they specifically talk about, those hair follicle stem cells. When we have a mature hair follicle, and this is a follicle that is going into the anagen phase, the epithelial cells and the mesenchymal cells are sending signals back and forth during its cycling. But what happens first? Who sends out the first message is the dermal papilla. And with that being the case, with that being the order of things, the stem cell bulge area that has those epithelial cells, they are not going to create dermal papilla cells. The dermal papilla cell identity is established earlier in the development from dermal mesenchyme cells. These are sort of intermediate cells that are transforming from their mesenchymal state and possibly to a intermediate population of a sort of soon-to-be-developed dermopapilla cell. So really, the structure that we care about the most that's renewing in each anagen cycle are the mesenchymal cells and the dermopapilla cells, because without them, we're not going to get any functional hair growth. So it's probably reasonable to think that we might find this cure somewhere in that mesenchymal layer to solve hair loss. When we look at PP405, there seems to be this assumption from the research that these hair follicles are normal and largely unaltered, at least outside of the epithelial hair follicle stem cell bulge region. Later on, we will talk about how this is not true at all, and it probably points to a major flaw in PP405 ever being an effective treatment. I have this suspicion that the Pelage and UCLA study that showed lactate dehydrogenase activation around the telogen to anagen transition could be describing a metabolic shift in the epithelial compartment as the hair follicle is preparing to grow. That kind of signal can help push a follicle that already has an intact responsive dermal papilla into anagen, and it does not necessarily address the separate problem of whether the mesenchymal compartment is competent or whether the dermal papilla cells need to be restored, expanded, or reprogrammed in a miniaturized follicle. It is also possible that the hair follicles chosen for the biopsy were already near the telogen to anagen transition, and the mitochondrial pyruvate carrier protein inhibitors that they used that are similar to PP405 in those mice, as well as PP405 in general in the human clinical trials, are mainly boosting activity in hair follicles that were already primed to restart to begin with. So they're not necessarily showing you that these hair follicles are dormant in the first place. They're just showing you a hair follicle that was already starting to grow, and maybe they amplified a particular signal. From my perspective, and again, I'm going to stress this is my perspective, I could be wrong, but when I look at the Pelage UCLA paper on lactate and hair follicle cycling, I find that the figures support the idea that the lactate and lactate dehydrogenase activity rise as part of an activation program that is already underway, rather than acting as the upstream step that creates and rebuilds the dermal papilla. So when I look at figure 1a, the telogen panel already shows some lactate dehydrogenase red staining in the bulge region, and the SOX9 staining is also highlighting that this is the bulge stem cell compartment. This could be a follicle that wasn't even fully dormant to begin with. And mind you, the signal becomes more obvious in the next panel, the telogen to anagen transition panel, and in the final anagen panel, while the SOX9 helps us confirm we are still looking at the same general stem cell bulge region as the follicle moves through these states. To me, that looks like a stepwise increase that tracks with activation states. It does not look like a clean on versus off switch that only appears once a fully dormant follicle is created again. Figure 1c and figure 1d also push me further in that direction because they show that lactate dehydrogenase genes and lactate dehydrogenase enzymatic activity increase during the telogen to anagen transition. And that reads like a metabolic ramp that accompanies stem cell activation. And it fits my interpretation that lactate dehydrogenase activity is part of the machinery that powers activation once the follicle is entering its growth phase. Figure 1e also supports that framing because the metabolic patterns shift in ways that are consistent with increased glycolysis and lactate-related metabolism during the activation in sorted hair follicle stem cells. Again, that feels like a stage change that comes with activation and not necessarily the first initiating event. The strongest functional piece in my view is figure 3. The deletion experiment shows that removing lactate dehydrogenase blocks hair follicle stem cell activation and keeps hair follicles from entering anagen normally. That tells me that lactate dehydrogenase is necessary for epithelial activation. But it does not answer the separate question that I care about, which is whether lactate signaling can restore or rebuild the mesenchymal instruction hub if the dermal papilla signaling is already compromised. And I should state this, that the dermal papilla cells are already functionally smaller and altered in androgenetic alopecia, and because the hair follicle unit gets smaller and smaller, it stands to reason that whatever is making the dermal papilla cells must also be impacted by DHT perhaps, or also something else along with DHT. The androgenetic alopecia gene has already been activated before the dermal papilla cells were created. In my view, I think the attention needs to shift to the specific signals that cause the mesenchymal stem cells to turn into dermal papilla cells. In theory, once we know that, maybe we can create some kind of treatment that can modulate those signals and create new dermal papilla cells. But also, more crucially, we need to know if these mesenchymal stem cells have already been reprogrammed to create faulty dermal papilla cells. Perhaps DHT and another signal already reprogrammed the mesenchymal stem cells along with nearby stem cells in the epithelium to produce signals that tell the mesenchymal stem cells to keep making faulty intermediate progenitor cell populations that would ultimately become faulty and diseased dermal papilla cells. So for now, let's assume that is the case, which later on we will go into some evidence that kind of points to that actually being the case. But again, let's assume that is the case. Then simply multiplying that mesenchymal stem cell doesn't really do anything if the stem cells have something wrong with them, because they'll keep producing diseased dermal papilla cells. Now, granted, you can probably find an area on your scalp that has healthy mesenchymal stem cells that produces healthy dermal papilla cells and therefore healthy hair follicles. And if that's the case, you can probably amplify that and, maybe clone the hair follicle organ and transplant it into other areas of the scalp, which we will kind of touch on later in the video as well. So just kind of, bear with me here. Perhaps some of these signaling cells may be found between the epithelial and mesenchymal layer. This layer is called the basement membrane, and keep this structure in mind because later on we will be looking at a study titled, quote, "The basement membrane of the hair follicle stem cells is a muscle cell niche," unquote, by Hironobu Fujiwara et al. 2011. So whatever this signal is to amplify and restore those mesenchymal stem cells, that would probably be the cure. So we need to find some kind of signal that can correct that mesenchymal defect and create better dermal papilla cells. And guys, I don't think that's going to be PP405. At best, it might shorten the resting phase of a hair follicle by increasing those epithelial stem cells to, start creating more hair sooner, like L'Oreal's P4H inhibitor stemoxidine, but it might not be as good as minoxidil, and again, that doesn't look like good news for PP405. We're also going to be looking at a study later on titled, quote, "Bald scalp in men with androgenetic alopecia retains hair follicle stem cells, but lacks CD200 rich and CD34 positive hair follicle progenitor cells," unquote. So please keep all of this in mind and definitely use those timestamps in the description below because we're going to get very in depth and I kind of want you guys to kind of, get my idea. So with that out of the way, we need to understand the exact activity that the dermal papilla is controlling in the hair follicle, and this is the hair growth cycle. You've probably heard of it, right? We need to know how this cycle actually works. The hair cycle explains why hair grows for a period of time, stops, sheds, and restarts the process all over again. Hopefully you get more hair growth and more cycles. Let's start with the anagen phase. Anagen is the growth phase, and early anagen begins when the hair follicle stem cells in the epithelial layer, they begin to produce derived progenitor cells, and those are intermediate populations. These progenitor cells start to proliferate, so they multiply, and they migrate downward to rebuild the hair-producing unit around the dermal papilla. Late anagen is when the hair shaft and the supporting layers are fully formed and the shaft elongates until it reaches its final length. Catagen is the regression phase. The lower portion of the hair follicle shrinks and the hair-producing structures collapse upward. Then, the dermal papilla pulls away as the follicle transitions toward rest. The telogen phase is the resting phase. During this phase, the hair follicle is relatively quiet and is not actively building a new hair fiber. The existing hair shaft is held in place as a club of hair. The dermal papilla during the telogen phase sits close to the stem cell bulge region, and this positioning matters because once it's time for the hair to start growing again, the mesenchymal stem cells will begin to reproduce and then ultimately form a new dermal papilla, and then that dermal papilla can restart the hair growth process by telling the epithelial stem cells in the stem cell bulge region to start growing again. So you kind of have this retraction-like effect where the mesenchymal stem cells are elevating towards the upper part of the skin structure so they can be close to the stem cell bulge area. And so when the mesenchymal stem cells are ready to reproduce proper dermal papilla cells, the new dermal papilla cells can potentially signal to the epithelial stem cells in the hair follicle stem cell bulge region to start creating those progenitor cells that create the hair follicle unit itself. After the telogen phase, you have the exogen phase, and this refers to the shedding phase of the hair. Now, shedding can occur in relation to the cycle, but it can also be somewhat independent in timing, which is why a hair can be shed even when there's a new cycle beginning right underneath that hair that is shedding. And finally, we have the kenogen phase, and this refers to a hairless interval in which the hair follicle is present, but the canal is empty, so there's no visible hair fiber emerging from the scalp yet. This is the dormant phase of the hair follicle. Once the right signals do occur, however, the hair follicle may transition into the anagen phase. And guys, this is where the defect kind of is. If the dermal papilla cannot produce strong enough signals, then maybe it just won't grow proper hair. And if the hair follicle stem cells in the stem cell bulge region are kind of compromised, then we're not really going to get, again, proper hair. So there's an interplay between the two. But the most prominent signal that coordinates all of this is the mesenchymal and dermal papilla cells. Androgenetic alopecia, however, is best understood as a progressive distortion of this cycle we just described. The growth phase becomes shorter, regression happens earlier, and the hair follicles spend more time resting or empty. Over repeated cycles, the hair follicle produces thinner and shorter hairs, which is the process called stepwise miniaturization. The key structure that helps explains why this is happening is the dermal papilla structure. Again, I've mentioned this structure so many times, but this is how important it is. And earlier in the previous segment, I've already told you that dermal papilla cells come from mesenchymal stem cells. Well, we can learn how important these mesenchymal stem cells are if we simply ask the question, what are they? Mesenchymal cells are connective tissue lineage cells, meaning that they come from the same broad family of cells that can form into tissues like bone, cartilage, tendon, fat, muscle support tissue, and marrow stroma. In the hair follicle, the mesenchymal dermal papilla cells function as a signaling hub that tells the epithelial part of the follicle when to grow and how to grow and when to stop. We've already touched on that before. And we've already touched on the fact that the dermal papilla signals to the stem cell bulge region that has those epithelial stem cells how to grow the hair. So a natural question should follow up. What is going on between the mesenchymal stem cells and the dermal papilla? What could possibly be causing these cells to create poorer quality hair? Well, if you're going to talk about the way cells grow and how they signal growth, we should probably look at a pathway known as the Wnt and beta-catenin pathway. When Wnt signaling is active, beta-catenin is allowed to accumulate inside of the cell. This protein, known as beta-catenin, can enter the nucleus and partner with transcription factors, such as TCF, to turn on growth-related genes. This pattern supports anagen, the growth phase, and supports the ability of the hair follicle to build and maintain hair-producing machinery. When the Wnt signaling is inactive, beta-catenin is actively removed. A group of proteins, often described as destruction complex, marks beta-catenin for breakdown, and the cell's protein recycling system, known as proteasomes, degrades the beta-catenin protein. So in simple terms, when you turn off the Wnt pathway early or when it's off indefinitely, the growth messages do not reach the nucleus, and the growth programs are less likely to stay on. If this is happening in the dermal papilla, that means 1, the dermal papilla is probably not going to get to a sufficient size, and 2, the dermal papilla is probably not going to be sending signals long enough to tell other parts of the hair follicle unit to grow hair. And really, this is exactly what happens to dermal papilla cells. The androgen DHT shifts this balance in genetically susceptible scalp hair follicles. This happens when free testosterone is converted into DHT by the enzyme 5-alpha reductase. DHT binds to the androgen receptor inside of the dermal papilla. after binding to the androgen receptor, the androgen receptor DHT complex recruits proteins known as coactivators and then moves inside of the nucleus. Once inside of the nucleus, the androgen receptor binds to specific DNA control mechanism regions called androgen response elements. And this is the crucial step where hormone signaling ultimately gets converted into altered gene expression. So, essentially, the DHT androgen receptor complex is changing the behavior of the cell. Because remember, your cell has your DNA. It has an instruction of all the proteins it should create and when it should create them. But this DHT androgen receptor complex is essentially telling your cell, hey, start producing more of these proteins that are ultimately going to make the hair follicles shift into a sooner telogen phase, that are ultimately going to make the hair follicles become smaller and more brittle, and ultimately probably not grow hair at all. So let's get a good overview on what those proteins are. First, we have DKK1, also known as Dickoff protein 1. And this is a protein that inhibits the Wnt signaling pathway. So here, in androgenetic alopecia, DKK1 activity increases. This is a problem because outside of the cell, there's something called Wnt proteins. And you also have a receptor called the LRP frizzled receptor. Now, in order for you to initiate the beta-catenin, remember, beta-catenin is a protein that tells your Wnt pathway to keep doing what it's doing, to stay on, and make the hair follicle keep growing hair. It relies on a Wnt protein to attach to the LRP frizzled receptor to have that sort of consistency. However, as DKK1 increases, it competes and interferes with Wnt proteins for that same receptor, the LRP frizzled receptor. Once DKK1 binds to that same receptor as the Wnt protein, then you're going to get fewer signals that tell the hair follicle to grow. And over time, as DKK1 increases in its output, the dermal papilla ultimately spends more time being off than it is on, and that means less signals, and ultimately that means less overall signals for hair growth, which is tantamount to a shorter anagen phase. Then we have TGF-beta1, and TGF-beta1 is a signaling protein that promotes regression-type behavior in the hair follicle. Higher TGF-beta signaling is associated with catagen-like changes, meaning the hair follicle is more likely to shut down growth earlier than it should. Then we have interleukin-6. Interleukin-6 is a signaling protein often associated with inflammatory-type responses. In this context, it can contribute to a less supported growth environment and can reduce healthy proliferation of the epithelial cells that build the hair shaft. So you're going to have more microinflammation around and even in the hair follicle. This can lead to weird immune responses, which we will touch on later in this video. So, if your dermal papilla cells have the genetics for androgenetic alopecia, then these outputs influence surrounding hair follicle populations. So, as the Wnt pathway signal decreases, this would mean a reduction in signaling that would otherwise convert hair follicle stem cells into those keratinocytes. And those keratinocytes are the cells that are actually building the hair fibers. They're building the hair shaft. And there are probably different kinds of keratinocyte cells that are responsible for building different parts of the hair shaft, the outer root sheath, the inner root sheath, and again, other important structures of the hair follicle. If this is compromised, this means we're getting less hair shaft fiber, small hair production per follicle, and even potentially issues with the skin barrier surrounding the hair follicle. Again, keratinocytes are going to make up the skin as well. So where do these keratinocytes come from? Well, keratinocytes are epithelial cells, so that means they have to be coming from the hair follicle stem cells. And those hair follicle stem cells are in the stem cell bulge compartment of the hair follicle organ unit. We also have fibroblasts and other supportive cells that shape the local skin environment. When the dermal papilla signal favors Wnt and beta-catenin activity, keratinocyte proliferation is stronger, so the keratinocytes are multiplying, but also the keratinocytes are bigger, and we can have longer anagen phases with thicker, healthier hair shafts. But as the dermal papilla signals inhibitors or blockers like DKK1, TGF-beta1, interleukin-6, the keratinocyte multiplication or proliferation, right, the number of times the keratinocytes can duplicate, that slows down. And cell death and regression programs increase and the hair follicle enters into catagen earlier and also rests longer. So the dermal papilla cells, once again, that are produced become fewer in number and also smaller in size, meaning the proliferation and growth signals are suppressed. And it all points back to those pesky dermal papilla cells. And if we want to go further, it points back to their origin, right? Something is happening to them when they're mesenchymal stem cells. Something is happening over there. So we have to figure out what is happening. But still, let's have a look at the dermal papilla cells and how they alter in androgenetic alopecia. We can look at a study titled, quote, "Androgen receptor-mediated paracrine signaling induces regression of blood vessels in the dermal papilla in androgenetic alopecia," unquote, by Deng et al., where the dermal papilla cells in balding scalps show increased nuclear androgen receptor localization. So what does this mean, right? In the areas of your scalp that are balding, if you have the condition androgenetic alopecia, those dermal papilla cells have higher androgen receptors because for whatever reason, the disease process forces those dermal papilla cells to create more androgen receptors. And for that matter, those dermal papilla cells want more DHT to destroy itself. It's a very, toxic kind of process. But in that way, if you think about androgenetic alopecia as a disease, of course, it's a defect. With this being the case, the dermal papilla cells are more susceptible to androgenetic alopecia because let's assume all of the follicles are sensitive, right? The ones that have more androgen receptors are going to possibly come in contact more with DHT. There's going to be more doors in which DHT can knock, can enter, and also activate those genes that are responsible for killing the hair follicle over time. Another thing they also noticed was that there was increased 5-alpha reductase activity in those dermal papilla cells. Now guys, increased 5-alpha reductase activity means that there's more instances in which testosterone can be turned into DHT. So it's not simply just having more androgen receptors is going to cause hair loss. No, not at all. But it's also in combination of having higher 5-alpha reductase activity, the enzyme that transforms testosterone into DHT. Those two things together are pointing at those dermal papilla cells being more susceptible to androgenetic alopecia. It's that combination. So the increased levels of DHT, again, alone isn't enough to further hair loss. We need a third part of the equation. And finally, that part of the equation is literally what your genes encode for. If DHT in that particular cell is activating DKK1 more, or interleukin 6 more, or TGF-beta 1 more, that means that, well, it's trying to kill your hair follicle. The researchers are showing that for those dermal papilla cells that are high in androgen receptors and 5-alpha reductase enzyme concentration, they tend to be in regions that are balding. And the hair follicles that are created in those balding regions, they lose their internal microvessels compared to non-balding hair follicles with functioning dermal papilla tissue. The study links the dermal papilla tissue structure. The study we have here is linking the dermal papilla tissue structure's vascular regression to DHT and androgen receptor signaling, which is a paracrine interaction. Paracrine, again, means hormones and their activity in a certain area. There is also TGF-beta signaling, which promotes apoptosis of nearby endothelial cells within the dermal papilla tissue structure during the early stage of the follicle miniaturization. We witness, once again, that the dermal papilla cells and tissue structure shrink over time. These are cells that are making weak signals to the stem cell bulge, which create weaker and thinner hair fibers. Over many cycles, this repeated bias toward early shutdown produces miniaturization. So essentially, if your dermal papilla are too weak or too small, they can't produce that many signals. And that means you're not going to get that much, signaling that tells the hair to grow longer hair, to grow thicker hair. The lack of signals reduce the strength and caliber of the hair. This is why in androgenetic alopecia, it's not simply stem cells being inactive or active. It includes a signaling problem in which the dermal papilla cells have something wrong with their development when they were just mesenchymal embryonic baby cells. Some sort of interaction, some sort of reduction in a particular protein or whatever it may be, which we will explore later, caused for these next generation of dermal papilla cells in the subsequent hair cycles to essentially change what they instruct the hair follicle organ to do. If the dermal papilla can no longer deliver strong, sustained pro-growth instructions, the hair follicle cannot reliably build a full-size anagen architecture. And guys, it doesn't matter. It doesn't matter if we increase what the stem cell bulge is doing, which is what PP405 claims to do. That doesn't matter. Those hair follicle stem cells can be stimulated all you want, but if the dermal papilla can't effectively tell them what to do, you're not going to get any sort of effective hair growth, if any at all. You're not going to get those hair follicle stem cells to differentiate, to transform into other parts of the hair follicle that would grow hair. I keep saying this thing over and over again, but again, guys, you have to understand how fundamental this is. Most of hair loss research is focusing on the wrong group of cells. This is why we don't have a cure. Previously, we talked about how a hair follicle goes through its hair growth cycle and the particular structures that are involved, being the dermal papilla and the stem cell bulge. Now, in this section, we're going to focus on the latter end of the hair growth cycle, just about when the hair follicle is about to recycle itself and go back into the anagen cycle. So it's the telogen to anagen transition phase. So when a hair follicle is ready to exit the rest phase and begin a new growth cycle, so going into anagen, the process does not immediately start with stem cells randomly waking up, but it starts with instructions from the derm papilla, the mesenchymal command center at the base of the hair follicle. During telogen, the dermal papilla sits close to the bulge region, as we said before, where the hair follicle stem cells reside. At the appropriate time, whatever that time may be, the dermal papilla releases a coordinated set of signals that tell those epithelial stem cells in the bulge region that it's time to initiate a new hair cycle. In response to these signals, various signals, the bulge stem cells do not immediately become hair-producing cells. Instead, they first step into a deep dormancy and generate a population of cells known as the early progenitor cells. These progenitor cells begin to multiply and then migrate downward away from the bulge and toward the base of the hair follicle, going towards the dermal papilla. As these progenitor cells migrate, they recognize the follicle's lower architecture, and they build the outer root sheath, reestablish contact with the dermal papilla, and ultimately give rise to matrix cells that produce the hair shaft itself. This downward migration and amplification step is essential. Without it, the follicle cannot transition from the rest phase into the productive anagen growth phase. So, in this case, you have a progenitor cell population. They are the cells that come out of these hair follicle stem cells that are from the epithelial layer. These progenitor cells represent intermediate cell populations that can further turn into various epithelial lineages required to rebuild the hair shaft, as I discussed before, right? The matrix cells, the inner and outer root sheath cells, and other components that are epithelial in nature that build the hair follicle. So, in other words, normal hair regeneration requires 3 linked steps. 1 being dermal papilla signaling, 2 being hair follicle stem cell activation, particularly we're focusing on the stem cell bulge, and finally 3, successful progenitor expansion and migration away from the stem cell bulge and down towards the lower architecture of the hair follicle unit. If any one of these steps fails, well, then you're going to see that the growth stalls. So we need to appropriately identify where these processes are breaking down in androgenetic alopecia. Let's first look at these progenitor cells that are coming and migrating away from the stem cell bulge region. These are the epithelial stem cells. In androgenetic alopecia, this signaling and migration pipeline appears to be disrupted. We can look at a paper by Dr. Luis Graza of Johns Hopkins University, as well as Dr. George Katsouleris of the University of Pennsylvania, titled, quote, "Bald scalp in men with androgenetic alopecia retains hair follicle stem cells but lacks CD200 rich and CD34 positive hair follicle progenitor cells," unquote. And in this paper, they demonstrated that in a bald scalp, the bulge stem cells, those epithelial stem cells, right, are largely preserved, meaning the reservoir of stem cells are still there. However,
the populations of early progenitor cells, so what comes after those cells are differentiated, are significantly reduced. Particularly, they seem to lack the markers of CD34 and CD200. So what is CD34 and CD200, and how come this finding is particularly important? Well, CD34 and CD200 are basically ID tags on certain cell populations in the hair follicle stem cells. They're not the hair itself, we have to make that clear, but they're markers that researchers use to track which cells are present and what stage those cells are in. CD34 positive cells represent the stem cells that have successfully responded to the dermal papilla signals, and in hair follicle context is commonly used to identify an early progenitor or early activated stem cell derived population in the bulge or outer root sheath lineage. So to put it simply, CD34 positive cells are often a sign that the hair follicle isn't just sitting with its own stem cells, but it's actually going towards the next wave of intermediate cells that expand and migrate downward to help rebuild the lower follicle during the telogen to anagen transition. So to put it very simply, it just marks a unique population of stem cells that are different from how they previously were. So early signs of differentiation. But for CD200, this is a surface molecule that functions as an immune identification signal. It helps the hair follicle maintain something called immune privilege. Now, immune privilege basically tells your immune system that certain cells and structures should not be targeted and attacked. So, if you have a sufficient CD200 along with other markers, then the follicle microenvironment is less likely to trigger an immune attack or inflammatory disruption right where the regeneration needs to happen. So, in plain terms, CD200 is part of what helps the hair follicle stem cells not suddenly get eliminated or killed by your immune system. So it gives these guys a chance to kind of amplify or turn into whatever cells they have to turn into to actually build the hair follicle itself. So this is why when Graza and Kotsouleris found that the epithelial hair follicle stem cells are still present in bald androgenetic alopecia scalps, the problem was now moved away from what happens after these cells are trying to differentiate. And what they noticed, again, was that CD34 positive and CD200 rich cell populations, the ones that the hair follicle stem cells are turning into, those are greatly reduced. And the implication is huge because this is pointing in the direction of the hair follicle having trouble doing the next step. And that's turning those hair follicle stem cells into stable, functioning progenitors that are literally responsible for making the hair. We can presume that if the CD200 rich cells are reduced, that means that the microenvironment may be less protective and more hostile. So you might have early progenitor cells that are less likely to survive long enough to complete that migration in order to rebuild the hair follicle. Maybe at some point your immune system sees that, hey, this thing doesn't have a sufficient amount of CD200, so it kills that particular hair follicle stem cell. On this micro level, androgenetic alopecia may have an immune pressure component, but it's not necessarily a classical autoimmune disorder like alopecia areata, but a state where immune privilege is weakened and low-grade inflammatory signaling becomes more permissive right at the hair follicle stem cell niche area where the regeneration is supposed to happen, thereby making the hair shaft and other hair follicle structures smaller, weaker, and more thinner. I like to think of this like type 1 diabetes. I think that serves as a good analogy for what it looks like when the source cells may still exist, but the environment prevents durable recovery. So, again, I'm not saying type 1 diabetes is related to androgenetic alopecia, but in type 1 diabetes, the target organ is the pancreas, specifically the islets of Langerhans, where the key functional cells are the insulin-producing beta cells. Early on, many patients still have residual beta cell function, which you can see clinically as measurable C-peptide. But because of the autoimmune process that targets the beta cells, that residual population has trouble expanding or recovering. Sure, the pancreas is still there, but the cells that are needed to regulate blood sugar levels are reduced, if not destroyed, because their early populations have been targeted by the immune system. Again, I'm not saying androgenetic alopecia is literally type 1 diabetes,
The point is the pattern. If the hair follicle stem cells loses immune calming signals like CD200, then the bulge where these hair follicle stem cells are and the early progenitor compartment, and again, remember this, these are the cells that come after the hair follicle stem cells have transformed in some way. Those cells in this particular state are likely to have some severe issues and be eliminated by the immune system. The environment becomes very hostile for them to proliferate and go to the next step, whether that means they turn into a matrix cell, etc., etc. That could mean that you're going to have more cytotoxic surveillance, more inflammatory cytokine exposure, more oxidative stress signals that right when the progenitors are trying to expand and migrate, boom, they get eliminated. So this finding pretty much contradicts everything Pelage has told us about PP405 right now. Even if a treatment increases proliferation markers like Ki67, that only tells you that the hair follicle stem cells are dividing, but it does not prove that the hair follicle is restoring the specific intermediate progenitor cell populations. Again, that CD34 positive that you can likely only get with proper dermal papilla signaling. It doesn't say anything about those progenitor cells getting a supportive immune privilege niche feature like CD200 that makes regeneration stable. So in other words, in androgenetic alopecia, the fragile link is often not can we activate something, but can we rebuild the whole regeneration pipeline? Can we get proper cellular proliferation and differentiation where the cells turn into what we need when we need them to build the hair follicle? So when you see Pelage Pharmaceutical talking about, hey, yeah, we've increased proliferation markers of the hair follicle stem cells, they're not answering any of these crucial problems. And I think it's because the lactate readouts that they had in their previous studies, it didn't prove anything. Maybe those studies are kind of suspect because, for all we know, the biopsy they took out, that telogen follicle that was reacting, it wasn't dormant. It could have been in a very, very early state of anagen. There's also another important structure that we have to talk about, and this is the Arrector pili muscle, what it is and why it matters. According to Professor Nilofar Torkamani and Dr. Rodney Sinclair in their article titled, quote, Androgenetic Alopecia, How It Happens, unquote, published in the Hair Transplant Forum International 2015, the Arrector pili muscle is a small band of smooth muscle that runs from the hair follicle to the adjacent upper dermis and epidermis. This muscle seems to be pretty important because it contributes to thermoregulation and sebum secretion. Another thing to critically consider is that the Arrector pili muscle arises proximally at the hair follicle at the bulge region. That's where the epithelial stem cells are. This point of attachment does not seem to be incidental. The bulge region, as we've noted many times before, is home to multipotent stem cells that are responsible for regeneration and maintenance of the hair follicle across its cyclical phases of growth, anagen, regression, catagen, or rest telogen. So the fact that anatomically speaking, the Arrector pili muscle maintains its attachment to this specific region throughout the hair follicle cycle suggests that there's some kind of relationship between the muscle and the stem cell compartment that is functionally significant and not merely anatomical. So there might be a mechanical function of this Arrector pili muscle. Perhaps it's involved in the contraction process, right? So what do we remember? Just a quick reminder, right? When the hair follicle is going into the resting phase, the telogen phase, the area that had the dermal papilla moves upward to be situated next to the stem cell bulge region. And we think the reason for this is so that the mesenchymal stem cells, when they produce new dermal papilla cells, they can properly signal to the hair follicle stem cells, those epithelial stem cells, to properly create new hair. And this allows the hair follicle stem cells, which are epithelial in that bulge area, to migrate easier and over time build the hair shaft and kind of displace the dermal papilla. So as it's building, it's like pushing the dermal papilla back down into the deeper layers of the epidermis. So that could be a very important function. And because it seemingly helps regulate this migration process, then that means the Arrector pili muscle must be very essential for hair growth cycling. It should be noted that the Arrector pili muscle does not simply attach to individual follicles in isolation.
The scalp hairs arise from follicular units that comprise primary follicle, right? So there's one follicle that's going to produce many hairs, possibly. And the Arrector Pili muscle and sebaceous gland, along with multiple secondary follicles, they arise distal to that Arrector Pili muscle. The muscle, therefore, serves as a structural organizer of the follicular units, physically connecting the follicles that share a common piliosebaceous architecture. The organizational role has been confirmed by multiple studies demonstrating that all follicles within a follicular unit share an Arrector Pili muscle. This is kind of confusing, right? Essentially, what's being said here is that you can have multiple hair follicles, each with their own dermal papilla, but then for some hair follicles, they could have one Arrector Pili muscle that joins them all together to make, let's say, 3 hairs. But also, each of those follicles can produce a number of hairs on their own. So you start getting very kind of intricate, complex sort of hair follicle layouts, where some people have a hair follicle unit that can produce, let's say, 2 hair follicles, and each of those hair follicles have like 1 or 2 hairs. It's pretty complicated. Anyway, the question of how the Arrector Pili muscle establishes and maintains its precise attachment to the bulge was addressed with the remarkable clarity by Fujiwara et al. In their study titled, quote, "The basement membrane of hair follicle stem cells is a muscle cell niche," unquote, published to Cell in 2011. This investigation revealed that the attachment is not a passive structural association, but rather an actively maintained molecular interaction mediated by specific extracellular matrix, proteins, and integrin receptors. Fujiwara et al. Demonstrated that the bulge stem cell deposits a protein known as nephronectin into the basement membrane. This deposition is highly localized, and it creates a zone of molecular specificity at the bulge that distinguishes it from the rest of the hair follicle's outer sheath. So this nephronectin protein serves as a ligand, and a ligand is simply a molecule that is specialized to connect to a specific receptor. Here, that ligand is for the integrin receptor alpha 8 beta 1, which is expressed on the mesenchymal cells, including the progenitor cells of the Arrector Pili muscle. So there's a cell population that creates the Arrector Pili muscle. But here's what's interesting, because that cell population seems to be coming from signals from the epithelial layer. So it's the hair follicle stem cell bulge that releases this protein that signals to the mesenchymal cells to turn into the cells that would ultimately become the Arrector Pili muscle. But the question is that stem cell bulge where the hair follicle stem cells are, is it sending out that signal itself? Or is that signal being sent out by the mesenchymal stem cells, which ultimately are becoming these early dermal papilla cells that send out this signal to the to the stem cell bulge epithelial layer that ultimately sends the nephronectin protein to actually create the Arrector Pili muscle? That is a lot of rambling, but it goes to show how complicated and integrated these signaling pathways are. The researchers are showing us here in this study that the alpha 8 integrin positive epidermal cells respond strongly to an environment rich in nephronectin. Once these cells are exposed to nephronectin, they upregulate smooth muscle cell markers such as alpha smooth muscle actin and smooth muscle protein 22 alpha. This means that the bulge stem cells are not only providing an anchor point for the muscles, but are actively inducing smooth muscle differentiation in neighboring mesenchymal progenitor cells. So this is the case where the hair follicle stem cell region, these epithelial cells, are influencing the mesenchymal layer through the basement membrane. The researchers confirmed this functional importance of this interaction by using some gene knockout experiments. So they got a bunch of mice, and then they removed the gene that was responsible for creating the protein nephronectin. The number of Arrector Pili muscles were reduced. Similarly, the deletion of the alpha 8 integrin receptor removed the ability for the Arrector Pili muscle to properly connect to the stem cell bulge region. What makes this finding particularly significant is the demonstration that nephronectin is a direct target of the Wnt beta-catenin signaling pathway.
Wnt signaling is one of the most fundamental regulators of hair follicle development, stem cell maintenance, and hair cycling initiation. Fujiwara et al. Showed that epidermal beta-catenin activation upregulated with nephrosectin expression in the bulge area, and as a consequence, as you had more nephrosectin, there was also increased alpha-8 integrin receptor expression in adjacent dermal cells. So what we're seeing is an increase of nephrosectin, which causes for the actual muscles to be created, and then you're also seeing the increase of other receptors and specialized proteins on the hair follicle bulge, which causes for the Arrector Pili muscle to kind of attach itself to that area. So this is describing a regeneration process of the Arrector Pili muscle. So with that in mind, how come there's a sort of clinical relevance of the Arrector Pili muscle in androgenetic alopecia? Because in androgenetic alopecia, it is often said that the destruction of the Arrector Pili muscle marks a point of no return in hair follicle biology. Essentially, that if you have an Arrector Pili muscle and it becomes severed, there's no way for you to properly get any sort of hair growth. Because remember, it seems that the Arrector Pili muscle is very important for lifting the mesenchymal layer of your tissue structure that has the derm papilla that would ultimately signal for the stem cell epithelial bulge to start creating new hair. And we largely get this understanding from the study titled, quote, Destruction of the Arrector Pili muscle and fat infiltration in androgenetic alopecia, unquote, by, once again, Dr. Sinclair and Torkamani et al. This investigation used three-dimensional reconstructions of serial histological sections to compare Arrector Pili muscles in patients with androgenetic alopecia, telogen effluvium, and normal controls. So what does this mean for people who don't understand? Well, they did a biopsy where they went into the scalps of various people with either androgenetic alopecia, telogen effluvium, alopecia areata, and people who don't have any hair disorders. They took out these hair follicles and then they created a 3D reconstruction digitally of the tissue itself. And they had some very interesting findings. So in all 8 androgenetic alopecia subjects examined, the Arrector Pili muscle has undergone degeneration and was being replaced by adipose tissue. So pretty much muscle was turning into fat. And once again, remember, these cells in the basement membrane that creates the Arrector Pili muscle, these are kind of mesenchymal cells in nature. And what we remember from mesenchymal stem cells is that they can turn into muscle cells, right? But also they can turn into fat cells. But really, what's going on here? Because in alopecia areata, in telogen effluvium, those guys, they're not having any sort of issue with their Arrector Pili muscle. The Arrector Pili muscle is still there. In the people with the healthy hair, the Arrector Pili muscle was once again still there. But the tendency for adipose tissue was noted in androgenetic alopecia. So something is causing for the mesenchymal stem cells that built the Arrector Pili muscle to transform into fat cells and not into muscle cells. And ultimately, the muscle cell kind of atrophies and becomes severed and replaced with fat cells, adipose cells. So these correlations suggest that the muscle degeneration is a progressive process that tracks with follicular miniaturization. The more miniaturized the follicle becomes in androgenetic alopecia, the greater the loss of muscle and the more extensive the fat infiltrate. Some residual muscle remained attached to terminal follicles in all androgenetic alopecia specimens, but the Arrector Pili muscle had completely lost attachment to the vellus hair follicles in all cases. The telogen effluvium subjects, once again, when you look at their biopsy, they didn't have fat infiltrate. I'm trying to remember right now, but I think it was like 3 out of the 5 had fat infiltrate, and the other 2, they only thought they had telogen effluvium, but when they looked closer, they actually had early androgenetic alopecia. So if anything, that kind of shows that fat infiltrate, or the tendency for a muscle to turn into fat, is very important for the pathophysiology of androgenetic alopecia. So the researchers walk away with the conclusion that telogen effluvium and alopecia areata are considered reversible forms of hair loss, whereas end-stage or advanced androgenetic alopecia is largely irreversible.
But it's not necessarily due to the severing of the erector pili muscle, but rather the erector pili muscle is changing. It's becoming something that it wasn't before, and that change is indicative of something. So the severing is not the cause, but it is a symptom of something else. And we know that simply severing the erector pili muscle can't really be the cause of hair loss, because in hair transplants all the time, we sever the erector pili muscle. And when we take the healthy hair, the donor hair, and we put it into the recipient area, so long as the recipient area is healthy and doesn't have any sort of inflammation, then the erector pili muscle rebuilds itself, right? And the hair follicle can once again grow. So that sort of signal points to something outside of the erector pili muscle being the defect, right? Something else is causing for the erector pili muscle in androgenetic alopecia to turn into fat. Some sort of improper signaling, right? And that could be between the mesenchymal layer and the epithelial hair follicle stem cell layer. And that could be it probably might be, the dermal papilla cells aren't signaling the right signals, for lack of a better term, to properly regulate the hair follicle. And over time, things spiral out of control and ultimately collapse. But let's revisit what we do know, right? From Fujiwara et al., we learned about nephronectin. And it seems that the nephronectin is secreted by the hair follicle stem cells, those epithelial stem cells, and they cause for the specialized mesenchymal stem cells in the basement layer to build the erector pili muscle. So it could be the case that the lack of nephronectin deposition from the hair follicle stem cells over time will cause for the muscle to turn into fat. And really the only thing that I can think of possibly causing this would be a lack of dermal papilla signaling. And we actually have some evidence here, right? Because Sinclair and Turkamani describe the Jaoda model in which the dermal sheath cells are lost from the follicle during the catagen or telogen phase, prompting the dermal papilla cells to migrate out to replace them. This progressive depletion of the dermal papilla results in a smaller dermal papilla, which in turn produces a smaller hair. The cells from the dermal papilla and the dermal sheath are capable of undergoing both smooth muscle and adipose tissue differentiation in vitro. So essentially what they're saying here is, hey, something's happening to these dermal papilla cells, maybe in their early sort of proto-mesenchymal state, where they're turning into fat cells for some reason. They also draw parallels to conditions such as Duchenne muscular dystrophy and age-related sarcopenia. In these disorders, you're essentially having the muscle turn into fat. So now, guys, we've identified various issues, and really, I've been trying to record this in parts because it's very, very long, and there's so much information to take into account, but we can do a quick sort of conclusion here, okay? What seems to be the defect up until this point of the video? We've identified that we need the mesenchymal stem cells to turn into the dermal papilla cells, and the dermal papilla cells need to have the proper integrity to signal to the epithelial stem cells that are housed in a component of the hair follicle organ known as the stem cell bulge or the stem cell niche bulge. Either or is fine. The dermal papilla cell, when it sends out a signal to these epithelial hair follicle stem cells, these hair follicle stem cells want to migrate out of the stem cell bulge and move towards the lower architecture of the hair follicle, right around where the dermal papilla is. From there, the hair follicle stem cells, they differentiate into various types of cells. Matrix cells, keratinocyte cells, outer root and inner root sheath cells, etc., but all of them are lineage from the epithelial stem cells. Keratinocytes are just one of these cells, and they help build out the tissue structure, the scaffolding of the hair follicle, and also the hair fiber itself. Fujiwara et al. Shows us that the protein nephrosektin is secreted from the stem cell bulge of the epithelial layer of the hair follicle. Nephrosektin deposits into the basement membrane and attaches to the mesenchymal stem cells that are there, and those mesenchymal stem cells ultimately turn into the erector pili muscles. And this is done by attaching to the receptor alpha 8 beta 1 integrin. But in order to have this complex machinery working, you need mesenchymal stem cells that are properly creating dermal papilla cells. Look, we know for sure that something is definitely happening with the dermal papilla cells. When you look at somebody's scalp with androgenetic alopecia, if you look at the balding areas and you examine the dermal papillas there, those dermal papilla cells are smaller. They typically have more androgen receptors. They also typically have more 5-alpha reductase expression. With that being the case, they have high DHT signaling to the dermal papilla. That shows us that DHT is causing for these things to shrink, right? Well, at least DHT might have its involvement in those dermal papillas. But it's conceivable, right? We have to think about this because these dermal papillas are smaller in every cycle. So something is happening to it before it becomes a dermal papilla. Something is happening to the mesenchymal stem cells. That's where the focus should be. And we have some very, very groundbreaking literature that we have to string together because I don't think people are doing this on a wide enough scale. To be fair, there are companies that are focusing on this, like Hair Clone in the UK, but we need to point something out. And I want to give a special shout out to one of my subscribers, and he's a good friend, a good pal of the KWRX Cognita server, and he pointed out some things. We've been going back and forth talking about the mesenchymal layer of hair loss biology. Guys, in this next section, we're going to get into the possible defect with the mesenchymal stem cells that cause for there to be androgenetic alopecia issues to begin with. So pay attention. This might be, I don't know if it's groundbreaking or whatever, but it definitely is something to be noted. So, is DHT the only cause of hair loss? Well, primary cause of hair loss. Of course it is. But it kind of gets a bit more complicated once you start looking outside and looking at the mesenchymal stem cells. So, let's get started.
So we've already established that the hair follicle runs on a conversation between the epithelial cells and the mesenchymal cells, with the dermal papilla being of mesenchymal origin acting as the command center at the base of the hair follicle, instructing the bulge stem cells, which are epithelial in lineage, to wake up and differentiate into hair-producing cells at the start of each growth phase. This mesenchymal and epithelial crosstalk is a replay of embryonic hair follicle development. And when that breaks down, you get miniaturization. The question is, what's actually causing it to break down? Well, here's where things start to get interesting. According to Dr. Simic Limbu's 2022 PhD thesis titled, quote, Investigation into Frontal and Occipital Scalp Dermal Signatures to Identify the Cause of Follicle Miniaturization in Androgenetic Alopecia, unquote, the dermal papilla cells from the front of the scalp, being the area that goes bald typically, are inherently less inductive than the dermal papilla cells from the occipital region, being the back of the head. She tested this using what's called a double sphere model. Essentially, you take dermal papilla cells and the bulge stem cells. You form them into a 3D cluster in a petri dish, and then you see whether those dermal papilla cells can instruct those stem cells to differentiate into hair-producing keratinocytes. It's a way of mimicking what happens at the start of each new hair growth phase, just outside the body. And what she found that was of particular importance was that the frontal dermal papilla spheres had diminished the ability to trigger that differentiation compared to the occipital dermal papilla spheres. So even in a controlled lab setting, stripped of all of the hormonal environment of the scalp, the frontal dermal papilla cells couldn't do the job as well as the occipital dermal papilla cells. So you have a question here, and that's why. What's different about these dermal papilla cells? Well, when Limbu did transcriptomic profiling of patient-matched frontal and occipital dermal papilla cells, she found the two populations have inherently different gene expressions. So in other words, they're not the same type of cell just sitting in different locations, right? Granted, they're both dermal papilla in nature, but rather they're functioning differently. Their genes are making them behave differently. Oh, of course, yes, they're growing hair or they're helping to signal grow hair, but the genes that are activated
are completely different. And when she drilled into the genes that specifically defined the occipital derm papilla cells, and those were the ones that were resistant to balding, she identified only 6 signature genes. And 4 of those genes were already known for contributing to androgenetic alopecia. Along with another condition, we're going to look at a specific gene known as TRPS1. TRPS1 is a transcription factor, meaning it's a protein that regulates the expression of other genes. It sits on chromosome 8 and has a well-documented role in hair follicle development, bone formation, and kidney development. In humans, mutations that knock out or reduce TRPS1 protein manufacturing cause a condition called trichorhinophalangeal syndrome. And this is where the clinical picture gets really interesting because multiple case studies show us what happens when TRPS1 is disrupted, and the pattern of hair loss that results looks strikingly familiar. It looks like androgenetic alopecia in some cases. We should bring our attention to the paper by Seitz et al. Titled, quote, Trichorhinophalangeal Syndrome Type 1, Clinical and Molecular Characterization of 3 Members of a Family and 1 Sporadic Case, unquote. In this paper, we're dealing with 4 patients that were described across 2 families. Let's start off with the sporadic case. The sporadic case was a 26-year-old woman who had never been able to grow her hair longer than 10 centimeters. She presented with thin scalp hair, non-scarring diffuse alopecia, with accentuation in the frontal parietal regions. She had a receding frontal temporal hairline and also some hair loss in her eyebrows. Mutation analysis identified a single base deletion at position 2110 of the TRPS1 gene, leading to a frame shift and premature stop of translation. Basically, a chunk of the genetic code got scrambled because one letter was deleted or inserted, which threw off how the cell reads everything after that point. The cell hits a false stop sign way too early and thus produced a shortened or broken version of the TRPS1 protein that can't do its job. The woman's parents were tested and neither of them carried the mutation. So this was a de novo event, meaning that it arose spontaneously in her. For the other case, which was a familial case in the same paper, involving a mother and 2 of her daughters, all carrying an insertional mutation at position 2480 of the TRPS1 gene. The younger daughter, 22 years old and 148 centimeters tall, had diffuse alopecia, a receding frontotemporal hairline, and, notably, her medial-occipital hairline, which was the hair in the nape of the neck, also started to recede towards the scalp in a triangular shape. She complained of extremely slow hair growth, about 1.5 centimeters over 4 months, with the maximum hair length around 8 inches. Her 25-year-old sister showed milder alopecia with a receding frontal parietal and triangular medial-occipital hairline. Their 51-year-old mother had the most subtle presentation, discrete diffuse alopecia with fine hair, a receding triangular medial occipital hairline, and, for whatever reason, this study notes protruding ears, which also seems to be a characteristic of some people with this disorder. All 3 carried the exact same mutation, yet the severity of their loss had varied dramatically. So maybe there's another modifying gene or protein that could be modulating it, which is why one of the, sisters has it worse, the mother doesn't have it that bad, and then the middle sister, she's somewhat okay. I think one thing that's interesting about the Seitz and colleagues paper is that they actually stress this particular point. They note that the TRPS1 mutation has a high penetrance but a variable expressivity. So essentially that means if you have the mutation, it's going to show up somehow. That is the penetrance part. But how bad it hits you varies wildly from person to person, even within the same family, as we've seen with the familial case from the case study. But the colleagues also suspected that many cases of trichorhinophalangeal syndrome go undiagnosed because the most disturbing sign for the patient is usually just having sparse hair. And if the cranial, facial, and skeletal features are subtle, then this disorder is going to get missed. It should be noted that this disorder is going to affect your bones, your muscles. It's affecting many forms of cartilage in your body. Anyway, going back to the case study, when they biopsied the scalps of these patients, they found something important. The number of hair follicles was only slightly reduced, and there were no signs of inflammation or scarring. What stood out was that the hair shafts had markedly reduced diameters, many showing a continuous thinning of the distal part with tapered tips. And you usually see this with uncut hair that simply can't grow long enough to require actual cutting. Another very, very interesting part, and I want you guys to really pay attention to what I'm about to say, was that the dermal papilla appeared to be hypoplastic, meaning it was shrunken relative to the well-developed hair follicle stem cell bulge region in the epithelium. So what's going on there? We have an underactive dermal papilla cell region, but we still have that hair follicle bulb or bulge that houses the epithelial hair follicle stem cells. That unit is still healthy. Well, Seitz and colleagues concluded that the impression of sparse hair in trichorhinophalangeal syndrome is mainly caused by marked thinning of individual hairs and not by a major reduction in follicle numbers. And that's exactly what happens in androgenetic alopecia. Miniaturization, but not necessarily destruction. But while we're at it, let's look at another study. According to the paper by Jian et al. Titled, quote, Trichorhinophalangeal Syndrome Type 1 Clinical Microscope and Molecular Features, unquote, we follow a 39-year-old Korean woman with short stature at 149 centimeters, who visited a clinic for fine, sparse hair and slow-growing hair with a receded medial occipital hairline of roughly triangular shape that had been present since infancy. She also had a receding frontal temporal hairline, rarefaction of the lateral eyebrows, a typical pear-shaped nose, and also the elongated philtrum. So really, this shows all the signs of somebody with trichorhinophalangeal syndrome. We have the classical skeletal issues, being that very short stature. We also have the facial structural issues as well. When they did an analysis of her genes to see where the mutation was, they found that a transition of cytosine to thymine at position 1630 in exon 4, resulting in amino acid change, put a premature stop of the gene translation. So in other words, there was some kind of change to the nucleotide, which causes for the protein, in this case TRPS1, only being partially completed, if not being created at all. When they had a histopathological examination of her scalp, it showed atrophic pileosebaceous units. Essentially, the hair follicles were shrinking. And they also noticed that there weren't that many hair follicles to begin with. And all of these issues presented without inflammation or scarring. However, on a scanning electron microscope, her individual hair shafts showed neither reduced diameter or structural defects. So in her case, the problem was more about the follicle loss than the hair shaft thinning. Once again, we can see that TRPS1 mutations can produce a range of hair pathology, from miniaturization to actual hair follicle atrophy. But again, it's going to depend on what is actually being disrupted in that particular gene. So one can have a defect with the TRPS1 gene protein, but the specific mutation is what's going to determine what happens to their hair and also what happens to their body. Once again, we can look to another study on the matter. According to the paper by Cho et al., titled, quote, Treatment of Hair Loss in the Trichorhinophalangeal Syndrome, unquote, a 17-year-old male patient presented with hair loss and slow growth since childhood. This particular male had all of the signs of trichorhinophalangeal syndrome. His aesthetics are as follows: bulbous pear-shaped nose, long philtrum, thin upper lip, and brachymetatarsia, involving the fourth metatarsals on his feet. And this is a condition where the metatarsals, and I'll try to put a picture up here somewhere, are abnormally short. The patient's alopecia was classified as a M3V1 on the basic and specific scale. This is a particular scale that we talked about on other videos. It's kind of like the Hamilton-Norwood scale, but it's a bit more specified, and you can get different sort of combinations that can describe thinning.
The team performed RNA sequencing comparing his balding and non-balding scalp and confirmed that the TRPS1 was downregulated in the balding areas. They tried 5% topical minoxidil for 6 months, and that didn't work. His hair loss progressed to a C3V1 with even more delayed hair growth. They switched to finasteride 1 mg daily for 2 years, and that didn't work either. His alopecia advanced to U1, which is essentially a unified pattern of loss. So he had a severe amount of hair loss during this time. Eventually, they transplanted 1,300 follicular units from the occipital area to the frontal and mid-scalp. The transplanted hair grew successfully and was maintained over the 3 years of follow-up, and the patient was satisfied with the results. So, good on him. Hopefully, he's having a better life. But one thing matters here, something really important matters here, and that is the occipital donor hair survived in the frontal scalp, just like you would see in cases of androgenetic alopecia hair transplants. This suggests that even in a systemic TRPS1 deficiency, the occipital hair follicles in some cases retain enough residual function to survive hair transplantation. The companion paper by Kim et al., titled, quote, Comprehensive Transcriptome Profiling of Balding and Non-Balding Scalps in Trichorhinophalangeal Syndrome Type 1 Patient, unquote, went deeper into the molecular picture of the same patient we just talked about. So it's going to describe the RNA process that they engaged in to see what was going on with the balding area of the scalp versus the non-balding area of the scalp. So when they extracted RNA from his balding vertex area and the non-balding occipital area and ran high-throughput sequencing, they identified 1,242 differently expressed genes between the two sites and validated 15 of them by quantitative PCR. So they basically took tissue samples from those balding areas and non-balding areas and saw the 15 most prominent important genes using a very precise instrument, kind of showed where the defect was. So the healthy hair follicle in the occipital region is the control, and then we have the, what we're trying to figure out, the balding, sick hair follicle is obviously the one we are assessing. 12 were downregulated in the balding scalp, and this included keratin genes like KRT6C and KRTAP3-1, and the cell proliferation marker MKI67. We also saw downregulated in the Wnt pathway, as well as a reduction of, you guessed it, the gene involved for making the protein TRPS1. The genes that were upregulated in the balding scalp included HBA2 and GAL. What I find to be pretty cool is that the paper is making conclusions that the pattern of hair loss and how it occurs in trichorhinophalangeal syndrome is very similar to that of male pattern baldness or androgenetic alopecia. And also, they note that TRPS1 has been associated with androgen pathways in prostate cancer and breast cancer. So why might they make that comparison? Well, it seems to be the case that in those diseases that are slightly dependent on androgen receptor activity, once DHT or another hormone like testosterone binds to the androgen receptor, it causes for there to be a change in the TRPS1 activity, and thus that can possibly make those cells act differently. So it stands to reason that with a defect like androgenetic alopecia that depends on an androgen receptor and DHT, then we're probably seeing something with this sort of protein that is being malformed in the mesenchymal stem cells, which is probably leading to hair loss. Interestingly, they also compared their gene expression patterns to those of a previous study on androgenetic alopecia by Graza and Kotsularis. We covered that paper earlier. The genes KRT6C and GPRC5D were downregulated in balding scalps in both conditions, and HPA2 was upregulated in both conditions. However, the prostaglandins, being prostaglandin D2 synthesis, which has also been implicated in androgenetic alopecia, was not significantly changed in the TRPS patient. And that might be a meaningful distinction because it suggests that TRPS-driven hair loss and androgenetic alopecia
share a common downstream architecture but might diverge at some level when it comes to certain androgen mediators. So now let's go back to Limbu's thesis. She found that TRPS1 was expressed at higher levels in intact occipital dermal papilla cells compared to the frontal dermal papilla cells. When she performed the knockdown of TRPS1, meaning she used a small interfering RNA sequence to silence or suppress the gene's expression, she did this in those healthy occipital dermal papilla cells. And when this happened, the expression of another gene called BLCAP also dropped. Now, BLCAP stands for bladder cancer associated protein. Okay, don't get freaked out. Just because you have hair loss doesn't mean you have bladder cancer, although that is a serious illness. So go get checked anyway. But still, the BLCAP protein is a tumor suppressor gene that regulates cell growth and apoptosis. And just because it's being expressed in the hair or other parts of the body, again, doesn't necessarily mean it's causing bladder cancer cells to be created in those hair follicles. No. You should just treat it as a protein in this context. So this protein regulates cell growth and apoptosis, and that is the process of programmed cell death. In the context of the dermal papilla cells, BLCAP protein was one of the occipital signature genes, meaning its expression is part of what defines the occipital dermal papilla cells as a distinct cell population. The fact that silencing TRPS1 caused for BLCAP to drop suggests that TRPS1 is sitting upstream, regulating part of the gene network that maintains the dermal papilla identity. So, if you lose TRPS1 and essentially don't have a functioning TRPS1 gene, and the dermal papilla starts losing the molecular characteristics that make it occipital, meaning that makes it resistant to hair loss, then it looks like the hair starts to miniaturize and starts to resemble the hair in the front of the scalp that was balding. So she made a very, very foundational discovery here. This is a brilliant PhD thesis, and I'm kind of losing my voice while I'm talking, so please bear with me. More critically, when Limbu silenced the TRPS1, she also did something else that was pretty cool. When she silenced the TRPS1 in the occipital dermal papilla stem cells, and then went on to test whether or not they were able to trigger those hair follicle stem cells in the dermal papilla region, again, that's of epithelial origin, she saw that the keratinocytes weren't able to differentiate. In plain terms, the occipital cells became less capable of signaling to the hair follicle stem cells those epithelial, again, I have to stress it, these are epithelial cells. The dermal papilla could not communicate to them to start making hair. And that is the functional link. TRPS1 isn't just a marker of the non-balding scalp, it's functionally required for the dermal papilla to do this in order to start growing hair. It needs this gene, and therefore it needs this protein. Now, we're going to get into the section where DHT might not be the only thing that's causing androgenetic alopecia. When it comes to the mesenchymal stem cells and how they turn into the dermal papilla cells, it looks like DHT might be having a companion in androgenetic alopecia. So we're going to get a bit mechanistic when looking at Limbu's study. Dr. Limbu also found that silencing TRPS1 increased the expression of EGR1, early growth response 1, which in her data set was upregulated in the frontal dermal papilla compared to the occipital. Remember, the occipital region is where we have less of the hair loss activity. EGR1 is overexpressed in prostate cancer and has been shown to bind to the androgen receptor and enhance its translocation into the nucleus. So, losing TRPS1 doesn't just weaken the dermal papilla's hair growing ability, it may also amplify the androgen receptor signaling, creating a feedback loop where the frontal dermal papilla becomes increasingly sensitive to androgens as TRPS1 expression declines. And here's where the androgen receptor enters in the picture directly. In prostate cancer, it's already been established that the androgen receptor represses TRPS1 expression. Limbu tested whether the same thing was happening in the dermal papilla cells. She treated both frontal and occipital dermal papilla cells with an androgen receptor agonist, and an agonist is something that's going to amplify a receptor's activity. So the one that she picked, it has a pretty weird name. I'm not even going to read it, but it's activating the androgen receptor similarly to how DHT would. In both cell types, TRPS1 expression was repressed. Then she added bicalutamide, which is an anti-androgen receptor blocker. In the occipital dermal papilla cells, TRPS1 expression recovered with use of bicalutamide, but in the frontal dermal papilla cells, the anti-androgen had no effect. TRPS1 expression stayed suppressed, and the frontal cells were essentially unresponsive to androgen receptor blockade when it came to rescuing TRPS1. And this has some pretty big implications because it suggests that the androgen receptor in the frontal dermal papilla cells may be functionally different from the ones in the occipital cells, possibly due to mutations or polymorphisms in the receptor itself. Limbu notes that certain mutations in the androgen receptor's ligand binding domain have been shown in prostate cancers to make cells resistant to bicalutamide. So maybe the same thing could be happening in these dermal papilla cells that are in the front of the scalp. Several polymorphisms of the androgen receptor have been associated with androgenetic alopecia. Whether similar mutations exist specifically in the frontal dermal papilla cells is unknown, but she did flag this as a critical area for future investigation. And I would have to probably agree with her assessment, right? I think there's something going on in the frontal dermal papilla cells, and even for that matter, the crown too, that makes them structurally distinct. We know from other studies that they have higher 5-alpha reductase enzymatic activity, they have higher androgen receptor density, so it probably reasons that these hair follicles are operating on a different program. Another thing that I want to note is that it seems like she's saying for the frontal dermal papilla cells that anti-androgens didn't seem to have an effect. I don't know if that's true, and a lot of people point this out in my study. When we look at people who undergo hormonal replacement therapy, particularly males transitioning to females, when they use medications like bicalutamide or they use high amounts of estrogenic compounds, some of the people that have severe hair loss, they grow their hair back to a significant extent. Maybe it says something about the baseline male TRPS1 signaling that once it's reduced, maybe higher amounts of estrogen can kind of blunt out the effects of DHT. So maybe the estrogen receptors on these mesenchymal stem cells could possibly upregulate TRPS1 activity in those males. And that could possibly be why you see a tremendous hair growth in those sort of transition cases. But you don't really see the same thing in people that are born with a female architecture. Maybe bicalutamide can help, but, injections of estrogen doesn't really help either. So female pattern hair loss that is androgenetic alopecia in nature might be closer to a greater TRPS1 defect than we realize. But that's my personal take. Now, let's zoom out to the population level. According to the paper by Perazzo et al. Titled, quote, GWAS for male pattern baldness identifies 71 susceptibility loci, explaining 38% of the risk, unquote. And this is a genome-wide association study of over 70,000 men identified TRPS1 as one of the 71 loci significantly associated with male pattern baldness. It sits at the locus of 43 on chromosome 8, with two distinct SNPs being single nucleotide polymorphism signals and a p-value of 2.7 times 10 to the negative 10. The study confirmed that the androgen receptor locus on chromosome X remains the strongest association, but it also revealed that the Wnt signaling pathway and apoptosis pathway play central roles, both which intersect with TRPS1 biology. TRPS1 is known to activate the Wnt inhibitors and regulate transcription factors essential for hair follicle morphogenesis. So the population genetics independently confirm that the molecular work is showing some kind of variation at the TRPS1 locus that contributes to baldness risk across the general population. So, in other words, TRPS1, when there's a sort of variation that occurs, it can lead to baldness. It can be a predisposing factor to baldness, and that doesn't just apply to people who have trichorenal phalangeal syndrome. It's also pretty cool to note that TRPS1 also finds itself in hair follicle biology in another way. According to the paper by Wahid et al. Titled, quote, Sequence and Structure-Based Assessment of Non-Synonymous SNPs in Hypertrichosis Universalis, unquote. Now, we're looking at a paper that talks about the various nucleotidic polymorphisms that contributes to hypertrichosis universalis. Now, hypertrichosis is excessive hair growth, and universalis essentially means all over. So, in other words, this is an excessive hair growth all over the body. TRPS1 was investigated, but not in the context of hair loss. In this context, it led to excessive hair growth. Mutations in chromosome 8 in the same region where TRPS1 maps. Dr. Limbu, in her thesis, pretty much points out the same thing. There are certain mutations of TRPS1 where a reduction in 90% protein activity has been associated with Ambras syndrome, a rare form of congenital hypertrichosis, again, resulting in excessive vellus hair type growth on the shoulders, face, and ears. So there is no binary switch where it's off and on when it comes to TRPS1. It's essentially, if there's a mutation, it can cause hair loss, but depending on where that mutation is, it may also potentially lead to hair growth. But it seems this is largely contextual based on what that hair is, right? Just because you see hair on your chest, on your arms, on the front of your head, and on the back of your head, they're all running different programs. And maybe in those mesenchymal stem cells that form the dermal papilla for those hairs, they all have a different TRPS1 behavioral type. So if there's a defect, it could essentially mean that, hey, the vellus hairs that are on my forehead that have been inactive since, I was born, that could possibly activate if TRPS1 signaling goes down. Whereas in the front of my hairline where I have excessive hair loss, if TRPS1 signaling goes down, well, that means I'm pretty much losing my hair. So I don't want to yap too much, but it shows something very crucial. What can I say about all of this? I can say that TRPS1 appears to be a critical gatekeeper of the dermal papilla identity. You need functioning TRPS1 in the mesenchymal stem cells. Functioning TRPS1 probably leads to better dermal papilla cells, which can effectively activate the bulge area of the hair follicle and get those hair follicle stem cells to migrate down towards the lower architecture of the hair follicle and ultimately create that hair we all love. Once TRPS1 signaling is lost, another protein EGR1 goes up, and that potentially amplifies androgen receptor signaling, and this ensues a vicious cycle that ultimately leads to the hair follicle that's affected by androgenetic alopecia collapsing. And that collapse is mostly happening in the dermal papilla and its mesenchymal origin. So, did I just come to the conclusion that DHT isn't so much the cause of hair loss as it's the activator of a deeper vulnerability? I guess you can say that. Still, DHT is a significant factor that we can control for, and really it might be the catalyst that's causing for all of this dysfunction to occur in the first place. But when it comes to this sort of discussion, is it the TRPS1 signaling? What is causing that signaling to become reduced? I just kind of walk away with more questions than I have answers at this point. But I do know that the actual susceptibility lies in the mesenchymal stem cells. That's where it is, and that's all I can reasonably say. So maybe if we can recover TRPS1 expression early, then we might be able to get those mesenchymal stem cells to become proper, and they can proliferate and potentially differentiate into stronger dermal papilla cells. So guys, we need some kind of treatment that can do this. And in the next and hopefully final section, so I can stop torturing my voice at 3:16 a.m. in the morning on February 28th, 2026. In this next section, we're going to be looking at SCUB3, Amplifica's whole lineup with AMP303, AMP502, I think, and 601. We're going to be looking at all that. Osteopontin, all of that research coming out of Amplifica. So let's get started. So to recap, in androgenetic alopecia, the damage is not limited to a single structure, but instead unfolds across multiple interconnected layers of the hair follicle. The dermal papilla, a cluster of mesenchymal stem cell-derived cells at the base of the follicle, acts as the command center for hair growth, progressively shrinking and losing its ability to send strong growth signals, as described in the previous studies we've looked at. At the same time, the Arrector Pili muscle, which anchors the bulge stem cell niche, again, this is the place that houses those epithelial stem cells, the hair follicle stem cells, this muscle degenerates and is eventually replaced by fat tissue. Again, a finding documented quantitatively by Torquemani and Sinclair in their three-dimensional reconstructions of androgenetic alopecia scalp biopsies. The basement membrane that sits between the epithelial bulge stem cells and the surrounding mesenchymal tissue is not just a passive barrier, but a dynamic signaling platform, as Fujiwara et al. Demonstrated by showing that the bulge stem cells deposit nephronectin into this membrane to recruit and maintain smooth muscle progenitors through Wnt-regulated signaling. When this basement membrane environment becomes disrupted, mesenchymal progenitors, again, these are stem cells that come after the mesenchymal has kind of differentiated. Those mesenchymal progenitors that should become smooth muscle or healthy dermal papilla cells may instead default to becoming fat cells, which is likely what we are witnessing in the progressive fat infiltration that Torquemani and Sinclair observed. Meanwhile, the epithelial stem cells in the bulge are still present even in bald scalps, as Graza and Cortsilarides showed. But the critical intermediate progenitor populations, marked by CD34 and CD200, are significantly depleted, meaning that the follicle retains its stem cell reserve, but lost the ability to convert those stem cells, those healthy hair follicle stem cells, into functional cell populations needed to rebuild a hair-producing structure. Here, there is likely an immunodysfunction component where many of these cells are deleted or suppressed by the immune system. Perhaps a CD200 agonist, like the one Arctis Biotherapeutics is creating, named ARQ234, could help, as it's designed to modulate the immune system in chronic dermatological conditions in order to help restore immunoprivileges. But you see, this is precisely the reason why PP405's approach appears to fall short, because it treats the bulge stem cells as though they're simply needing a metabolic wake-up call through lactate dehydrogenase activation, essentially assuming that the stem cells are the bottleneck and that boosting their proliferation will restart the hair cycle. Remember guys, proliferation and differentiation are two different things, right? You're getting more of something if you proliferate. To differentiate, you actually have to transform one thing into something else. But the evidence across these studies we've looked at suggests that the bulge stem cells, again, those are the hair follicle stem cells that are epithelial in nature, are not even the primary problem in androgenetic alopecia. They're still there and they're still capable of dividing. The real breakdown is happening downstream and around them in the mesenchymal compartment, where the dermal papilla cells have become smaller, fewer in number, and increasingly incapable to deliver a sustained Wnt-driven growth that signals to hair follicles that they have to enter and maintain a productive anagen phase. PP405 does not appear to address the progressive failure of the dermal papilla cells, the degradation of the basement membrane signaling environment, the loss of nephronectin mediated niche maintenance, or the depletion of CD34 positive and CD200 rich progenitor populations that Graza and Kotsularis identified missing in bald scalps. In my opinion, to be quite frank, PP405 is based on research concerning follicles that were otherwise healthy enough to use lactate as its driver. But studies like Pai et al.'s 2024 paper titled, quote, Activation of Integrated Stress Response in Human Hair Follicles, unquote, show us that with chronic abuse of this system, using a similar molecule to PP405 known as UK5099, which, by the way, the Pelage UCLA team used in their initial study when they were, discovering this pathway, by using these compounds, it led to hair follicles eventually not having enough energy, and this ultimately caused those hair follicles to collapse. Because as a hair follicle grows, and as any cell grows, and by the way, hair follicles are fast growing in terms of their cellular nature, so the energy demand becomes more amplified. Well, because they were using an insufficient source of energy, being lactate, to create ATP, eventually the follicles, the human hair follicles, mind you, entered into early catagen. And if not, they entered into something scary sounding called dystrophic anagen. And this is where the typical growth phase is suddenly stalled due to improper cellular energy management. PP405 is meant to be used chronically and applied to the scalp. It could end up causing the reverse issue where you suddenly pull all the other hairs that were otherwise healthy out of their growth phase and into a shedding state. If not, you could possibly permanently damage them. These are very sensitive pathways they're playing with. So all of this information tells us is that androgenetic alopecia is a disease that is kind of complicated. And by disease, I mean in the, histopathological sense. I don't mean like if you have androgenetic alopecia, you're going to die, right? But it is a dysfunction. But it's a dysfunction that affects various compartments, with its sort of primary focus being on the mutation of the TRPS1 protein, if not just the general behavior of the mesenchymal stem cells. The epithelial stem cells, those hair follicle stem cells in the bulge compartment, are perfectly fine for the most part. But it's about getting those proper dermal papillas to be created from the mesenchymal root. And if this can't happen, well, you're just not going to get good quality hair. But with that out of the way, I think the more realistic place to look forward to is how could we actually get this to work? How do we actually stimulate the mesenchymal stem cells to create proper dermal papilla cells? Do we have anything in the global pipeline of hair loss research medications and therapies and protocols? Do we have anything that could possibly address this? And guys, we kind of do. This is where I think Dr. Maxim Plikus' work gives a clearer path forward than just waking up cells. According to Liu, Guerrero Juarez, and Plikus in their paper titled, quote, Hedgehog signaling reprograms hair follicle niche fibroblast to hyperactivated state, unquote, hair growth is not only governed by epithelial cells, and we already know this from the previous studies. It's governed by niche fibroblasts that include dermal papilla cells, and shifting the state of those mesenchymal niche cells can overactivate hair growth through a downstream activation of a molecule called SCUB3 or SCUB3. In other words, SCUB3 seems to be a mesenchymal niche factor, this protein, that can push the entire follicle system toward growth when the niche is placed into the right state. And this matters because it aligns with the idea that the hierarchy of hair loss research should lean upstream toward restoring the mesenchymal behavior and the dermal papilla function, and not simply thinking about stimulating those hair follicle stem cells in the epithelial layer. A glycoprotein known as osteopontin is a multifunctioning protein that can modulate the immune system and also regulate how cells behave. According to another paper by Maxim Plikus and his colleagues Wang et al., titled, quote, Signaling by senescent melanocytes hyperactivates hair growth, unquote, hairy nevi, also known as hairy moles, like a skin mole, not the animal, the mole, but these hairy skin moles are made of senescent melanocytes, but they're able to produce a substance that leads to hair growth, and that substance seems to be osteopontin. So I should note that the key concept here is not that cellular senescence is good. It is that some senescent cells can secrete a strong signal package that can change behavior of neighboring intact cells. And that could be for better or for worse. But in that paper, osteopontin emerges as a dominant secreted cue, and the hair growth effect depends on the CD44 receptor signaling. Another thing to note is that the CD44 receptor is present on mesenchymal stem cells, and osteopontin-like molecules like to bind to it. So there is a possibility, given the research thus far, an osteopontin-derived molecule, like what Maxim Plikus' company is looking towards, being AMP303 or AMP300, may cause these cells to produce better dermal papilla cells. So it is conceptually interesting. Obviously, there needs to be further research to see if this is the case. However, it may be, considering how this is derived in moles that are already producing hair. We essentially have a potent, specific instruction signal that can push hair follicle behavior into a more active, regenerative mode through a defined ligand-receptor relationship. And that ligand-receptor relationship would be the osteopontin and the CD44 receptor. So these two proteins, these two compounds, SCUB3 and osteopontin, again, they're being researched by Amplifica. Amplifica's making their own versions of these. I think SCUB3 is AMP601, and the osteopontin analogs are AMP303, AMP205, and the other ones I think AMP501. Check if, I might be wrong on that, but it's something like that. That's their breakdown. So all of this is probably like 20,000 years into the future, right? Well, maybe it might take a long time for it to get to the market. Maybe not. But we do have clinical trial results from Amplifica with the use of AMP or AMP300, and it's presented in the paper titled, quote, AMP303 Injectable Treatment for Androgenetic Alopecia, a multi-center randomized placebo-controlled feasibility study of a novel polysaccharide, unquote. And it establishes that AMP303 or AMP303 has a sort of clinical efficacy that's been noted in a sort of early phase 1 kind of study. So this is really cool because we have a sort of randomized placebo-controlled nature that is validating the extent to which this thing can actually grow hair. So just to briefly explain the methodology, here 32 patients, all male, were injected with AMP303 to only one side of the scalp. In these patients, the injected side produced a statistically significant increase in non-vellus hairs. Another thing to mention was that they only did one injection in 6 months. Just one injection. So this may be some good news because it certainly prompts further research into AMP303 and also other osteopontin analogs that could possibly be created. And it also gives the AMP601 SCUB3 analog some more, bolster to it. That should be researched further. And it's pretty cool because each patient kind of serves as its own control, right? So one side hadn't been treated, but the other side, it seemed to have, localized effects and very, very specific localized effects. However, with all that being said, I think the research needs to come back to focusing on restoring the mesenchymal behavior and building functional dermal papilla cells. There are other companies like Hair Clone UK. In fact, that's where I got the Limbu study from. Dr. Simic Limbu actually works for Hair Clone UK, and they've done some excellent things, so hopefully they can keep expanding on this sort of research. So guys, that's pretty much it for this long video. Hopefully it's clear. We did try something out that's new with the editing, and yeah, that's it. Thanks for watching, guys, and I hope to see you in the next video. See ya.